Okay, alright. So, hello dears. Ayan. So, wow, muna siya ako siging intro, no? Okay, alright. Well, anyway, ayan. So, welcome yet again to another pre-recorded lecture in your clinical bacteriology class. And medyo hapit na gito man, gamay na lang. So, we're now starting again with the different, we're starting with the lectures on the different methods and the different ways on how we process our specimens, right? So we have started already for your swab specimens, your throat swab, and then next what we are now going to discuss is the stool, okay, are your stool specimens. So how do we process your stool specimens for culture? Now, for this lecture muna, before we start with our stool lecture proper, what we're going to start first, or what we're going to discuss first is the different culture media that we use for routine stool culture. So as you can see, di ba, medyo kato pa lang sa una, I've always mentioned yeah, I'll discuss the culture media natong gamiton for stool. So wala ko siya gi-appeal sa culture media prep kay para dili na siya para at least ma-correlate niyo siya dayon ba asa siya gamiton. Okay? And these culture media again are usually used for stool culture. So before we start with the discussion on stool culture proper, like how we process the stool culture, it is really imperative and it's fitting that we start first with the different culture media natong gamiton for stool culture. Okay? So PDF lang ni siya. Actually, handout pa na ko ni siya. Sa atong third year pa ko, no? Kaya wala ko nakapangayo sa ako ang previous teacher sa si Sir Jemu. So may ganin na pa ning PDF. Okay? So again, these are the culture media that we use for routine stool culture. Okay? Alright. So we'll start first with the first one is of course our best friend, the McConnellagar. Okay? This have been well gasgas na to sa discussion natin, di ba? We've always discussed about McConnellagar, and it's an enriched, it's a selective and differential medium. Now, for this lecture po guys, atong culture media for stool, we will talk about the different composition of the culture media, like iyahang indicators, iyahang kaning mga inhibitors, etc. And silang purpose, okay? So, again, we'll start first with McConnellagar. Again, it's a general purpose. It's for the isolation of your enterics, or the Enterobacteriaceae and other gram-neg rods. And actually, it's a primary plating medium. When you say primary plating medium, it's one of the first choice, o diba? Sana all first choice. It's one of the first choice na mga plating medium natong ginagamit. Okay, and that's McConnellagar. Alright, yung inhibitors, again, are bile salts and crystal violet. Because again, we want to make sure that in McConnellagar, ang mutubo or gyud is ang gram-neg bacilli, or ang gram-neg rods, especially your enterics. So therefore, we put inhibitors para ang ubang mga kagaw, especially the gram-positive cocci, dili motubo. Okay, and these are in the persons or in the presence of bile salts and crystal violet. So these are the inhibitors. So kani sila ang magpa-inhibit na mutubo ang gram-positive organisms. So kung nagani tubo sa McConnellagar, so you're very much certain and you're very much confident na mga gram-neg na sila, gram-neg rods. Okay? Alright, indicator unsa neutral red, kung acidic siya kay red, and alkaline pH is yellow, and the carbohydrate unsa lactose. Okay? Please take note. Now, again, muna siyang pink colony because again, di ba, your organism, if lactose fermenter, yung i-break down ang lactose to acids, di ba? Di ba the results of fermentation of sugars are your acids. So therefore, because of the acidic pH, may mo siyang red, and it will now appear as pink colonies dire. Di ba? Recall? Alright? So how do we, again, ang hirap pag hindi PowerPoint. So again, characteristic colonies or unsa tong lantawon sa McConnellagar, kung pink, dark pink, or red, again, these are your lactose fermenters, and kung colorless, non-lactose fermenters. Now, usually if you are really looking for pathogens, okay, katulad yung mga non-enterics or mga enteric pathogens, usually ang pinaka-significant good is the colorless, okay, non-lactose fermenter. If gikan sa stool, okay, we'll discuss that later sa atong stool culture na lecture. Okay, so but again, these are the different characteristic colonies na itong lantawon dapat for McConnellagar. Okay, so that's the first culture medium. Now, again, I have to emphasize, guys, na for this lecture, what you have to take away or what you have to learn good are the different components, okay, of the culture medium. Unsay sulod niya, unsay pH indicator na, unsay inhibitors, and unsang expected na mga colonies. Okay, please take note. Ayan, muna siya important ani na lecture. Alright, next one we have what we call your okay. So of course, pink, dark pink, or red, your lactose fermenters, EKE, Escherichia, Klebsiella, Enterobacter. Colorless and then pink, of course, these are your late lactose fermenters, usually after pila pa ka hours of incubation. So you have katong late lactose, Citrobacter, Serratia, Hafnia, okay? And the colorless, your non-lactose, Salmonella Shigella. Usually these are mga really pathogenic na enteric organisms. Okay, so muna siya itong gina-look after usually. Okay, again, colorless colonies. Okay, next culture medium is your sorbitol McConnellagar, as mentioned, again, SMAC. It's usually good. It's for the isolation of your EHEC or E. coli 0157H7 or enterohemorrhagic E. coli. And the carbohydrate there is, ang difference lang yun is ang carbohydrate, which is D-sorbitol. Ngano ganito? Because again, E. coli 0157H7 is a non-sorbitol fermenter. That is why ang significant na colonies sa SMAC are the colorless colonies because we are looking for E. coli 0157H7. Yes, gasgas na din. Palagi nang inuulit. Pabalik-balik, paulit-ulit na. Again, colorless colonies on your SMAC, E. coli 0157H7. And again, on sa atong culture media of choice for E. coli 0157H7, SMAC. Okay, so balik, sige, balik-balik rin ng mga questions. Okay, alright. And the rest of your E. coli strains, of course, they ferment sorbitol, so they appear pink pa rin on sorbitol McConnellagar. Okay? Alright. Now, if you look into the presentation and atong discussion po, the remaining or most of the remaining culture medium after McConnellagar are usually the culture medium used na good for the isolation of Salmonella and Shigella. Because again, these two are really considered to be enteric pathogens na good. The rest mo good sa ato ang mga like Enterobacteriaceae E. coli, mga Citrobacter, Kleb, mga inana, those are opportunistic pathogens. But under normal conditions, sila kayo sila makakause of infection. Unlike ani nilang Salmonella Shigella na good na medyo considered good sila na enteric pathogens. So if you'll notice, atong discussion after McConnellagar, the culture medium natong or culture media natong discuss, most of them, if not all, are usually for the isolation of Salmonella and Shigella. Okay, and we'll start with bismuth sulfite agar. Again, isolation of Salmonella. Inhibitors, bismuth sulfite and brilliant green. So these two again inhibit other organisms aside from Salmonella species. Okay, and the indicator nato is H2S because recall that Salmonella again is an H2S producer. So the ferrosulfate is the source, okay, of the sulfur para makaproduce ng H2S. And ang carbohydrate na to is glucose, alright? And this is your bismuth sulfite agar or BSA. Okay. Alright. Dili na siya Bachelor of Science in Accountancy. Charot. Okay. Alright. And kung black with metallic sheen, that's specifically Salmonella typhi. And black with light green lang, walay black, metallic sheen, that's other Salmonella species. Okay, because some of the other Salmonella species, if I'm not mistaken, di na sila produce H2S, okay, or light ra kaayo. But for black with metallic sheen, it's characteristic of Salmonella typhi. Okay, the next culture medium is also your Salmonella Shigella agar. Nasa pangalan na, SSA. So, wag na mag-isa. Para sa manang SSA, Salmonella Shigella. For the isolation of Shigella and Salmonella species. So, your inhibitors, bile salts, sodium citrate, and brilliant green. So, same ra sa BSA. Ang naadra is sodium citrate. Alright, indicators is pH, for pH is neutral red, so same sa imong McConnellagar. Alright, H2S ferric citrate, okay? And sodium thiosulfate pa rin as sulfur source. Same siya with your TSI, di ba? As the source of sulfur. Okay, alright. And the carbohydrate there is lactose. Okay, so again, this is your Salmonella Shigella agar. One of the culture medium dyan na ginagamit for isolation of Salmonella and Shigella, your SSA agar. Okay, Salmonella, Shigella, agar. Okay, and what are the characteristic colonies? For pink to red, it's a lactose fermenter, so other members of your enterics, no? Colorless, non-lactose fermenter. And last, if colorless pero na black center, non-lactose fermenter and H2S producer. Kagets na mo, usually if nandiyo mga black, or mga blackening, na black center, whatever, that signifies that the organism produces H2S. Okay? Alright, so kinsa ni silang mga lactose fermenters? Of course, kung, wala din na mention. Sorry. So, of course, kung pink to red lactose fermenter, other members of the enterics, Enterobacteriaceae, so kato na mga E. coli, other organisms. So, ang atong ginalato kanila number 2 and number 3 because colorless, non-lactose fermenter, pero dili mo produce H2S, that's characteristic of Shigella. And number 3, colorless, non-lactose, pero mo produce H2S, so na ay black center, that could be Salmonella. Because again, Salmonella produces H2S. Okay, alright. So, that's for Salmonella, Shigella agar. Okay. The next one is still another culture medium for the isolation of Salmonella and Shigella. They're XLD, xylose lysine deoxycholate agar. Still the same purpose, Salmonella Shigella species isolation. Ang inhibitor niya is sodium deoxycholate, which is a bile salt, all right, in lower concentration. So still inhibits usually gram-positive, okay, and other organisms. Usually gram-positive good. And then, imuhang pH is phenol red, the acid pH is yellow still, and alkaline pH is red. H2S is ferric ammonium citrate na precipitate. Kato di ang sa BSA, guys, sorry. So, katong sodium sulfate dito, muto siya precipitate. Okay, dili di ang source sa sulfur. Okay, sodium thiosulfate ang sulfur source dili sa XLD. Okay, alright. And ang precipitate para makita ng blackening is ferric ammonium citrate. Okay, alright. Ayan. You have the carbohydrates, tulo na kabuok, lactose, sucrose, and xylose, and ang amino acid, L-lysine. Okay? Alright, so that's for XLD. So, again, di ba? Actually, if I'm not mistaken, mo ba niya ang culture media of choice for Salmonella Shigella? I'll have to check. It's either naras SSA or XLD, pero I think ang XLD to siya. Alright? Basta, again, familiarize lang din sa components, unsay mga characteristic colonies. Okay? Alright, ayan. So, for, again, characteristic colonies, kung yellow, fermenter. Okay? Kung yellow with black center, fermenter and H2S producer. Kung red ra siya kay non-fermenter, okay, pero wala may black, so dili siyang produce of H2S. And kung red with black center, xylose fermenter, H2S producer, and lysine decarboxylase positive. So, you're detecting decarboxylation of lysine. Remember na ay lysine, okay? So, therefore, you're determining also if capable ba siya decarboxylation of lysine. Okay. And, for yellow, again, other intestinal flora that may grow ferment. So, mga E. coli, catocleb, etc., mga fermenters. Yellow with black, enterics na produce og H2S na fermenters, na produce og H2S. Example, Proteus, Citrobacter. They produce H2S, diba? Which may not have been completely inhibited. And of course, red, one of the characteristic or significant dyan nato is Shigella. Okay? Shigella do not ferment any of the carbohydrates and they appear red or clear or colorless, diba? Because Shigella is a non-fermenter of lactose, okay, and other sugars, okay. And red with black center, of course, Salmonella, not ferment lactose or sucrose, but it does ferment xylose, which is essential in decarboxylating lysine to revert the acid pH to an alkaline pH. So, sorry, okay. So, dili siya fermenter of other sugars, but mo ferment man siya xylose. And in the same time, iyang i-decarboxylate ang lysine. Okay? So, what happens is, ang acidic pH, may mo siyang alkaline pH. That's why mo color red, nagyapon iyahang color. Diba? Kay yellow man if acidic, niya red if alkaline. Because of the decarboxylation of lysine, diba? Those are mga amines and those are usually basic. So, because na-decarboxylate ang lysine, so from an acidic pH, because of na-ferment niyang xylose, diba? Nabalik siya to alkaline pH this time because of the products of lysine decarboxylation. Okay? Alright. So, red again with black center. What is just take note? Usually, Salmonella, if not the other enterics na produce of H2S, usually Salmonella, na colonies na yung mga black center, blackening of the medium, black metallic sheen, and okay? Alright. That's 4 XLD. Okay. Next one, we have Hectoan enteric agar. Still the same for the isolation of Salmonella and Shigella species. I like this culture medium kaya ganun ko siyang color. Okay, alright. Inhibitor niya is bile salts. Alright, indicators this time will change. Bronchithimal blue. Acidic pH is still yellow. Alkaline pH is blue. For H2S, it's ferric ammonium citrate day ang mo combine to form the precipitate, diba? And still sodium thiosulfate ang sulfur source. Alright, so same regapon with TSI. And the carbohydrates this time kay lactose and sucrose. Alright? Okay. And your characteristic colonies, bright orange to salmon. Okay, that's salmon. Salmon, silent L. Salmon pink. All right, lactose and or sucrose fermenter. Green to blue-green, non-fermenter. And then green to blue-green, nanay black na pod. Non-fermenter pero H2S producer. Okay, so as you can see again, bright orange to salmon pink. Lactose fermenters, that appear orange to salmon pink. So other members of the enteric, especially katong mga lactose fermenters. So what we're looking for is the green to blue-green and kaning green to blue-green na ay black center. Because again, for green to blue-green na walay black, because again, dili mo produce H2S, that could be Shigella. And green to blue-green na ay black center, that could be Salmonella. Okay? So muna siya, diba, kabantay mo na, kung Salmonella, usually na ay black na something sa iyahang colonies. Because again, it produces H2S. Alright. Okay. Next, we have, again, another, your EMB agar or eosin methylene blue. It's usually for a general purpose na for the isolation of the gram-neg enterics. Ang inhibitors niya is eosin Y and methylene blue. And the indicators is, muna siya ang culture medium na kung unsa yung inhibitors, mupod yung indicators. Eosin Y and methylene blue. Okay? And carbohydrates, lactose, and sucrose. So, eosin, methylene blue, or EMB, agar. Okay? So, again, general purpose na siya, maka-isolate siya gram-neg, usually for the gram-neg enterobacteriaceae. Okay? Alright. And again, yung inhibitors, same ra po sa indicators niya, eosin Y and methylene blue. Nasa pangalan na. Okay? Alright. And carbohydrates, lactose and sucrose. Alright. Characteristic colonies, blue, black with green metallic sheen, rapid lactose and or sucrose fermenter. Pink, lactose and or sucrose fermenter. So, dili siya rapid, like after pila pa ka-hours of incubation. And kung amber or colorless, non-fermenter. So, actually, eventually, same ra gyud ang results if non-fermenter ka or something kay di gyud mo change ang color, either colorless na ba or like walay change of color, di ba? Alright? And ang characteristic ra gyud ani for your EMB is the organism that produces your green metallic sheen, di ba? Ka-recall mo ani. Green metallic sheen, kinsa ni? On EMB, you have E. coli, o di ba? Si E. coli, green metallic sheen on EMB, E. coli. Alright. And another one is also fish eye colonies. Fish eye. Fish eye colonies, murag mata sa isda. On EMB, kinsa ni siya? Please take note, this is Enterobacter. Okay? Characteristic. Enterobacter, fish eye colonies. So, nasa murag mata sa isda. Okay? Alright. Ayan. Okay. And the last one, if I'm not mistaken, the last na nato is thiosulfate citrate bile salts, sucrose agar. Ayan. So, this is again the culture media of choice for the isolation of Vibrio species. Ayan. Please take note. So, we have the inhibitor sodium citrate, sodium thiosulfate, and 10% oxgal. Ang 10% oxgal, if I'm not mistaken, that is a bile salt man yata, Japon. Okay, and sodium citrate, sodium thiosulfate, again, inhibitors of the other organisms. Again, diba, this is considered to be the culture media of choice for Vibrio species isolation. Okay? And the pH na to is bromothiamyl blue or bromthiamyl blue. Acid pH is still yellow, alkaline pH is blue. H2S this time is ferric citrate. Okay? And ang sulfur source pa rin is sodium thiosulfate. Okay, so if I'm not mistaken, ang ferric citrate pa rin, ang point ani katong sa previous good nato na mga siya di ay ang mo combine to form the precipitate. Okay? So ang ferric citrate. If I'm not mistaken ha, I'll have to check. Okay, all right. And carbohydrate nato is of course sucrose. So na siya actually sa ngalan, guys. Thiosulfate citrate bile salts muna ang inhibitors. And then ang sucrose mo is sugar, di ba? So medyo para din na mo maglibog kung unsa ang inhibitors sa iyang sugar. Alright? And again, para kakinsa ni siya, for Vibrio. Wala na lain, guys. Vibrio, TCBS. Okay? Pero kung enrichment media of choice for Vibrio, of course, that's APW, alkaline peptone water. Okay. And again, please take note yung pH indicator, bromothiamyl blue or bromthiamyl blue. Ayan. Ay, sorry. Ang hirap talaga kung hindi PowerPoint. Okay, ayan. For characteristic colonies, yellow, of course, sucrose fermenter. Blue-green, non-sucrose fermenter. Yellow with black center, sucrose fermenter and H2S producer. And blue-green with black center, non-sucrose fermenter and H2S producer. So as you can see, I hope ma-correlate ninyo. Basta ganun yung mga black-black, is usually mo produce siya H2S. And then, sa different colors niya, either depende sa pH indicator, it could be na mo ferment siya or dili. Okay? So I do hope that mag-gets siya ang murag connection. Okay? Ayan. And for characteristic colonies, yellow, sucrose fermenting Vibrio such as si Vibrio cholerae and Vibrio alginolyticus. And kung blue-green non-sucrose fermenters, Vibrio parahemolyticus and Vibrio vulnificus. Okay, the rest na mga katong na black center could be na mga other enterics na na-isolate nato or nakuha from the specimen. But what we're looking for kan yellow or blue-green, kung yellow again could be sucrose fermenters, again Vibrio cholerae and alginolyticus. And kung blue-green, you have parahemolyticus and vulnificus. Okay? Alright, so that's for TCBS. And yes, so that's the end of our culture media for routine stool culture. So as you can see, I do hope medyo na-gets na kung asa ni sila gamiton. So ato na silang gitapok tanan because these culture media, although palagi na itong gi-mention from the previous lectures na to, earlier lectures, these culture media, ato na silang gitapok as one karon because again, they serve the same purpose, which is for stool culture and for the isolation of enteric pathogens, diba? So again, ang important lang yun ninyo i-remember ani is ang iyang composition, unsay ang inhibitors, unsay ang pH indicators, unsa iyahang characteristic colonies. Okay? Alright, so muna na siya for atong stool culture media na na lecture. Okay? So I'll now see you on our next lecture, guys. Okay? So have a great day, dears, and keep safe.